Welcome to solverfy.
Hi everyone, my name is Eric and let's focus on problem 44. So in this question, we are focusing on the concept of DNA isolation. And in the question, they are presenting us different scenarios and they want us to identify what is likely to occur. So let's walk through the general steps of DNA isolation. In DNA isolation, you start with a cell, and in the cell, you're going to have DNA. So that is the content that we want to obtain. Taking the cells, we're going to move it into a solution where then it's going to be placed into a centrifuge for lysis. So the centrifuge is going to spin the cells at a very high rotation per minute, and thus that rotation uses gravity to break open, and centrifugal force actually, to break open the cells. So that's called lysis. And when you're spinning it in a column, you're going to find that in the column, you're going to have liquid, and at the bottom, you're going to have the sediment called the pellet, right? So up here is called the supernatant, and down here, the red part is the pellet, which is the sediment, right? So that is separating the contents of the cell, and typically, the DNA will be found in the supernatant. And then once this has occurred, we are going to move the contents into a new column where we can purify and obtain our DNA. So let's run through each of the answer choices and figure out what is likely to occur. So with part A, it says that the DNA is going to separate into the supernatant, and that is true because we do find that after centrifugation, so that lysis portion, we are going to find that the DNA is going to be found in the supernatant, which is the liquid. Let's look at part B now. B is not true because DNA is not affected by a protease. A protease is an enzyme that degrades proteins, and since DNA is not a protein, it won't be destroyed by the protease. Instead, what's called a DNA ase is actually the part that degrades DNA, right? And then for RNA, there's a such thing as a RNase, right? Which is the enzyme that degrades RNA. So B is not true. Part C, DNA is unaffected by the RNA. While that might seem true, it actually isn't true because the RNA is going to be the contaminant when we're trying to purify and isolate our DNA. Therefore, DNA will be affected by RNA, and it is also not true. And finally, D, DNA combines with the RNA. That is not true because DNA does not interact with RNA in that respect. So, after considering all of our answer choices, the correct answer is A, where DNA is going to separate into the supernatant. I hope this helps, and thanks for listening.